Okay, part two. So you can also use the computer to measure changes in shape. And often a change in shape will be a change in outline. And so there would be ways to measure those sorts of differences. For example, the difference between a platelet and an activated platelet is that the regular inactive platelet is just sort of round, like a kind of an oval, like a bread roll, and the activated platelet goes all spiky and really cool looking. They would have the same mass that is still conserved. They're just changing the way that mass is distributed. And so you could look at, image a little section of blood, a sample of blood rather, and look at the platelets and gauge based on the shape the amount of activation there, for example. Yeah. There would also be things like texture and patterns that you can set your computer to look at and measure. A plowed field would have furrows, for example, which is something that would not occur in nature normally. Yeah, and you can, look at this. You can see how the land is divided into lots. You can see how the lots are being used for agriculture. And you clearly got different kinds of crops. Now, I don't know that you can identify which specific crop we've got here, but you can see that at least something is in a different stage of growth. And so, very likely, you've got farmers here that are rotating their crops. They're putting maybe the light green thing is the thing that gets them all the money. So they're loading up on those. But then maybe they're rotating through fields that are fallow, which you see here, little brown fields, or fields where they'll plant something that enriches the soil, like peas will do that, which may be some of these sort of darker fields. Yeah, could be a lot going on there. You could correlate this with other data. You could have survey data that says, okay, farmers, what are you growing in the area, or what is being sold at market after this? And you could then go back to the satellite images and say, okay, if it was 75% winter wheat, you can say, wow, 75% of the squares are this nice bright green. I'll bet that's what the winter wheat looked like at this particular time of year. And so, yeah, you can learn lots of things. When there are shadows, when the sun is coming in at an angle, then you can get height information. And if you've ever played around with Google Earth or Google Maps, there's actually a tool in there that'll let you look at things from an angle as if you were sort of flying over it, sort of add pseudo depth to the images, which is really quite neat. Yeah, and geologists like low sun angle, yeah, to see fault lines and stuff. And we'll actually do a little project during class where we use something like that. All right, so here you go. You can see lots of different buildings here, possibly even a university by the look of it. Some sort of little pond in the center. That's kind of interesting. All right, so the idea is that you get the digital images, and usually these will be purchased or they'll be made available from a government. Someone like Google buys them from the companies that put the satellites up there. Not anybody can actually afford to put the satellite up there, but the ones that do then will make their money back by selling these images to mapping companies that need this for the up to date image of what's going on. You then do pre-processing. If you were trying to analyze for a particular color, you could shield out all the other colors and just focus on that one. That's something you can definitely do in ImageJ. Pull out the features you're interested in, enhance those if necessary. Interpret, classify, yeah, all these things. It's one of these where I think you can't really say, oh, this is how you do image analysis, because every project is kind of different. You're treating an image as if it was numbers, and you're treating the image as if it is connected to other information that we know, like a list of addresses or something like that. you can't say there's just one size fits all. This is the way you do it. So this slide is trying, and full marks for trying, right? All right. There are areas that are not imaged well because the satellites are taking these kind of bizarre paths where they don't perfectly record everything. They may, get one area twice or three times before they get another area next to it even once. And so there's a certain amount of image joining, rectification, like patching up the missing bits. This can be done very nicely using AIs, among other things. There are other ways to do it that are less smart, but that would be good. And you've also got to look for geometric distortions, like if the satellite is, for example, looking out from an angle and seeing things a little bit sideways as opposed to right over the top. Or like in the previous example, it looks to me as though there's a little bit of fisheye going on here, maybe, as though everything's kind of curving out from the center. Maybe that's just an optical illusion for me. I'm not sure. So the process of image enhancement is improving the appearance. This can be useful if you're just trying to map lines. Like if you're trying to make a road map and then map sewers onto the road, you don't really care about slight differences in the pixel intensity of a road versus a sidewalk. And so you can put it in, use a thing called a sharpening filter, something like that, which will basically attempt to find a sort of a smooth, hard line that divides two areas as best it can. It's imperfect, it's altering the data, and if you're doing something where the image pixels are important, where those are your data, and it's not just the like mapping something, then there are only some kinds of image enhancement that would be useful. You could, for example, enhance by filtering out all the frequencies of light that you're not interested in. Yeah. And then you can make an image histogram, which has all the brightness values from 0, which is very, very dark, to 255, which is as white as can be. And you can see this sort of spread. And so you can then normalize this so that the brightest pixel is going to be 255. You can mathematically spread out the rest of the data. And so the darkest pixel is 0. And when you're adjusting contrast, that's essentially what you're doing. You're sort of spreading this function out or condensing it down or shifting it a little bit to the right or to the left if you want to make it brighter or darker, respectively. Okay, so I certainly like using 16-bit tagged image file format files, TIFF files. They'll usually end in .tif. There are 8-bit TIFF files. That's what normal civilians use. The 16-bit carries more precision in the exact grayscale tone, for example. And so you can make finer judgments based on more refined data, basically. Then you can normalize it, and you can normalize the different colors so that the reds are always, right on the middle of red, even if it was, I don't know, you get these times, if it's sunset and the sky is a little bit red, then everything that you can see is going to look a little bit red in the image, and so you can correct for that. And you want to make sure that there's an even playing field, basically. And then ideally, you would then do some sort of verification in the real world. Now, the thing that they are going to talk about over the next couple of slides is image analysis of a microarray experiment. So let's talk microarray. There are lots of different ways to do a microarray. What you're doing is putting different versions, of the same thing down in little spots, and you sort of glue the spots down. It's not glued, but effectively that's how it works. So that each little spot will have, for example, a different piece of DNA. And then if you want to see what a protein binds to, you can put all these different DNAs and lay them all out. You can actually have every possible combination, maybe, of DNA sequence on there, at least of a short sequence. And you can put your protein over and you can see, okay, where does the protein bind? If you're able to label the protein with some sort of color, or label the DNA in a way that the protein will block the light from that, then yeah, you can just basically take a picture of it and you can then interpret that. This is very much like what's going on inside the machines that are doing next-gen sequencing. Of course, right? Yeah, like those Illumina machines where they take a picture of a spot and they tell which color the spot is, exactly like this. Same thing with the pyrosequencing, where they're reading out the spots in each of those little wells and seeing how bright the light is. Yeah, that image analysis is actually done in a proprietary way. Proprietary just means something that the companies keep secret from us. It's operating by what are presumably fair principles. It's just that that's their intellectual property, how they actually do this. You can do it yourself. You can make your own array and you can interpret the array. You can build your own program. And then, hey, you can sell it to somebody else or make it free on the internet for someone to use. So yeah, you can scan for different colors separately. You can scan for all the colors together. Once you scan for different colors, you could combine them, and it's most modern papers, the style guides recommend that you don't use red and green. So if you're saying, what are all the gray dots on here? Yeah, no kidding. Yeah, these guys are red over here. These guys are green, and when they combine them, that one turns yellow because it is both red and green. So there you go. Amazing. But yeah, you can combine or you can break down the images into their different color components of your microarray. So examples of microarrays that I have done. If you have an antibody and you know the antibody binds to a protein, but you don't know which part of the protein it binds, that's easy. You can take the target protein, the one the antibody binds, and you can break it down into little tiny regions. Let's say, 15 amino acids at a time. And you can do 1 through 15, and 2 through 16, and 3 through 17. It's very easy to translate these and glue them down to a grid. And then, of course, you just wash your antibody over, maybe even fluorescently label your antibody, and you can tell exactly where your antibody binds and where your antibody does not bind. And in that way, you can tell what's the specificity of your antibody. Which part of the protein is it binding? What exact sequence? Stuff like that. We've also done it with enzyme activity. So if you have a sample from a cell that has perhaps some kind of disease, perhaps some kind of viral disease, you can, they make these substrates, which are chips, which are kinase substrate chips. So for example, there are serine threonine kinases, but they bind to a specific protein. And then if they have that protein with a serine or threonine, they will add a phosphate on. And so it's that kind of kinase. And so you can have an array with thousands and thousands of different things that can be phosphorylated. And you put some phosphate in the buffer, and you put your cell extract or your enzyme that you want to see, what does this do? And if it's a kinase, you will find out exactly what it's able to add phosphates to. Now, these experiments are kind of expensive. They'll run from a couple $100 to a couple $1,000 with analysis. So you would want to do some other bioinformatics before you just go and sort of slap a new protein on there and say, maybe it's a kinase. You would want to see some sort of other confirmation that it looks as though it might be a kinase. Yeah, you can do things like this. Binding, enzymatic modification, addition of particular modifications to glycosylation. All of these things exist as different microarrays. So they're pretty neat. So the way a microarray experiment works, you're going to have some kind of dye, which is going to highlight whether or not something has happened. So in the case of the kinase array, you'll put the protein on there. It'll either add phosphates or not, and then you can come over the entire chip with one antibody that binds phosphoserine or tyrosine, and another antibody that binds phospho. What the other one? No, serine threonine or phosphotyrosine. Yeah, there you go. All right. Yeah, and so the laser is going to shoot in some light. You're going to excite some sort of fluorescent thing. The photons are going to come out of that. Those are then going to hit some kind of screen, like a CCD, camera capture device, I think is what that stands for. And that is like what you have in your phone. It basically converts photons into energy, and the energy then shows up as a signal that goes to a chip. And so the chip then stores like a little array. It's like a little Excel sheet where there's a value in each of the little boxes. And that is the brightness or color or whatever of the image. Yeah, and you can have a couple values in there or make a couple maps. And then you can convert that and you'll get a signal. There you go. So here's an entire genome SNP mapping array. If you were to go to companies like Ancestry.com or 23andMe, I believe the way they currently work, and they may change this next week, they don't actually sequence your genome. What they do is they put you on a SNP chip. So SNP is a single nucleotide polymorphism. And so they have things that are complementary to different single nucleotide polymorphisms. Like if there's one version of a gene that has an A nucleotide in the middle and another that has a T nucleotide in the middle, if you have that little loop and you have DNA from a person, you can tell which one of these things it hybridizes with. And so you can see, oh yeah, okay, it hybridizes more with this one and less with this other one. Therefore, we can tell what your genotype is. Or if it hybridizes a bit to both, you can say, hey, you have a mixed genotype. And yeah, that can happen. Okay. Then you can basically calculate the intensity. In both of these, where you see like RFG, RBG, and GFG, GBG, that's the foreground, which is the area of interest. And because you know the way that the array was set up, you know that there's going to be a spot every, 100 microns, something like that. And so you can set the computer, you can just find the 4 spots in the corner, and you can tell it, okay, there's going to be 100 spots between these, 100 wide, 100 long, and they're in an exact array because they were laid down there, basically 3D printed onto this little chip. And so you can say, all right, how red is the spot that I'm looking at, and how red is the area around the spot? So you can measure the foreground, subtract the background. That'll get rid of any sort of, if there's a little red tint or if there's a bit of sloppy binding of whatever your red fluorophore is. And then you can find the ratio of the red to the green, and you can calculate that and come up with basically, what's going on. So one of the problems you'll occasionally see if you do a lot of these microarrays is that you'll have, see these are called comet tails. See how they all smear? Each smear is coming from a very bright dot. And so it probably means that the antibodies were flowing across and they seem to be sort of coming loose. They're like binding and then sort of bouncing along a little bit after they bind. And so even though this is the spot, you're ending up throwing a little bit of yellow onto this spot over here. You can get areas where something bizarre happens and the background is really high. Like look at that spot down there. That's crazy. But the foreground, which are the little spots, are almost black. You can get blotches. And a blotch is just going to be a place where, if you're reading this out with an antibody, or a little pool of that antibody binds. And so as part of the image analysis, you would need to set the computer to say, hey, only take spots of a certain size, or only take spots of a certain intensity, or, you sort of set up rules or only take the center of the spot, something like that. Like this one up here, it's actually 3 spots. It's just that the spots are spreading out as they're interacting with the sample. And so that's a practical problem that can occur. And there are some things you can do to correct this. But one of those, and probably the best one, is you're usually not going to have just one spot on the array for each thing you're trying to measure. You're usually going to build in redundancy, REDUNDANCY, which just means that you're going to have the same spots in multiple positions. So if something weird happens to the panel on the left, you've got an entire panel on the right, and you can read out multiple times for each experiment. And you can look at the variation. You can compare the intensity of the spot to the intensity around the spot, which would tell you whether your spot has sort of bled. Like something like this is just a very big spot, but you could look directly in between, like on the diagonal in between these. That would be a good spot to measure background right there. And you can see, yeah, that background is still nice and black, and so this would be potentially usable, whereas this would probably not, because the background spot would be right there. Okay, areas of high background, absolutely. If you're using antibodies for this or anything, even a Western blot, the thing to watch is that antibodies are sticky molecules. And they can stick even in places where they're not supposed to stick, because you're putting them on these weird metal and plastic and, protein arrays with very limited amounts of, water present at some times. And yeah, it's just not very physiological. If you think of antibodies, they're either in your nose or your gut or in your bloodstream. And if they're in your bloodstream, they're being pushed around really fast by a really strong current. And so that current, that flow, and all the other stuff in your blood, all that serum albumin, for example, protein, is going to make it very difficult. It's going to be competing with the antibody, so only a very specific antibody will actually bind and be able to hold on. Whereas in these experiments, you sometimes have to do it a couple of times to eliminate problems like this, because you can get the antibodies holding on where they really shouldn't be holding on, and it's just an artifact. You can get spot overlap, as you can see here. Here's some of these spots merging together. So maybe there was, too much rehydration, too much water during some of the processing, like where you put the antibodies on to make it show up. And you can end up with, there's a little piece of clothing fiber. You would be surprised how beautifully fluorescent some little threads from clothes are. And there are some of them that glow bright green or blue or yellow or white. And they're really beautiful, except when they're in your image, because they make it very difficult to actually get accurate numbers. You would probably want to exclude everything that this string is touching and use some of the replicates of the same array. So the two 16-bit images, the green dye that people use, or one of them, is called Psi3, and the red dye is Psi5. Those can be they're recorded separately and then compressed into images that only show the green and the red. You can throw away the rest of this. You can display the fluorescent intensities for both of them with an overlay image, and there you go. Represent the results. What people would normally do to be sort of friendly to people who are who see things differently is you would normally convert the green to blue, which you can do with an image analysis tool. If it's this bright green, you turn it into the same brightness of blue, and it's the same digitally, because you're just representing a thing. Same thing with red, you might turn that into yellow, and blue and yellow will come together, and those would make white when they do come together. So there you go. Here's the red dye. If you were looking at, what's the binding of my red dye tagged antibody versus my green dye tagged antibody. There you go. And you can then take the intensities And since we know the map, we can say, okay, if it's this green and this red, we can put that together. And so if they're all green, they're going to show up like that. If they're all red, they'll show up like this one. And if they're about evenly green and red, they'll show up here. If they're more red, they'll show up a bit orange, like all these guys, etc. All right, Image J, made by the NIH, or at least maintained by them. And you can download your own copy. Now, for the homework, you can also use, there's a free copy that's available that you can access through the internet without having to download and install this. But if you want it, it's a good image manipulator. You can do a lot of things that you can do with Photoshop with ImageJ. And it will also do quantitative things. It will turn pictures into numbers that we can then use to answer questions. Yeah. So there you go. And that's what the page looks like, or at least used to look like. That's an older version. And that's the end of image analysis. Hope you enjoyed it.